Welcome to educator.com. In today's lesson, we're going to be discussing organic compounds. And living organisms are made up of organic compounds. Organic compounds are compounds that contain carbon. Compounds that do not contain carbon are known as inorganic compounds. And the field of study of organic compounds is called organic chemistry. An example of an organic compound would be something such as methane, CH4, whereas water, H2O, is considered inorganic. Now, even though water is inorganic, it's obviously very important to biology and to life. So that's not to say that living organisms don't contain or need inorganic compounds, but they're mostly composed of organic compounds, and that's going to be our focus of today's lecture. A couple of exceptions though. Although carbon monoxide and carbon dioxide contain carbon, these two are generally classed as inorganic. So there's a couple other exceptions as well, but these are the important ones that contain carbon, but generally scientists refer to as inorganic. All right, starting out with the basics, organic compounds have a carbon skeleton. So a carbon skeleton is a backbone of carbon, and then the carbon atoms are bonded to other atoms. This carbon skeleton could be various lengths. It could be just a couple carbons long. It could be many carbons long. It could be branched. So it could have carbons attached to this first linear section of carbons. The atoms that are attached to carbon could be various atoms, but there's certain ones that are particularly important in biology and that we're going to keep returning to. And those are hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So these are the ones we're going to focus on, although, of course, living organisms do contain other elements and use other elements such as calcium or magnesium, trace elements such as iron that we discussed in a previous lecture. But for organic compounds, carbon plus these other 5 elements are the ones that you're going to see occur the most. Now, carbon is a very versatile element, and that makes it an excellent basis for biological molecules. Let's think back to the structure of carbon, and that will explain its versatility. Recall that carbon has 6 electrons. That means that it has 2 in the first electron shell, and that shell is filled. That shell only holds 2. It has 4 electrons in the second shell. So its valence shell contains 4 electrons, and that leaves 4 empty spots. 4 more electrons to get to a total of 8 for a full valence shell. Therefore, it needs 4 electrons to fill its valence shell. And it can fill that shell in various ways. It could form 4 single bonds with other elements, such as, say, hydrogens here, and then the other carbon next to it. So that's one way it could fill. It's sharing 4 electron pairs with other atoms. Another possibility is that it could form double bonds. So remember, with CO2, carbon forms 2 double bonds, one with one oxygen, one double bond with one oxygen molecule, and oxygen atom, and then another double bond with the second oxygen. That again gives a total of 4 total shared electron pairs, and it'll fill the valence shell of carbon. So this ability to share so many electron pairs gives carbon a lot of versatility and allows, just from a few different elements, a huge range of molecules that can be produced, and these molecules are the basis of life. Hydrocarbons are molecules that consist only of carbon and hydrogen. So here, this would be an example of a hydrocarbon. And the entire molecule is not necessarily a hydrocarbon. It could be a situation such as a protein, or actually other large biological molecule where there's a chain of just a section of hydrocarbons. So there could be a large biological molecule that has various different atoms on it, but one section of it is hydrocarbon, and that hydrocarbon section is going to be nonpolar. So hydrocarbons are nonpolar, and they form regions of molecules that are nonpolar as well. So you could just have a hydrocarbon, you could have a larger molecule with just a hydrocarbon section on it. We're going to focus on 4 classes of organic compounds today. Actually 2 today and then 2 in the next lecture, but overall 4 important classes. But before we do, we need to go on and discuss the concept of isomers. Isomers are molecules that have the same molecular formula, but they differ in their structure. And there are 3 types of isomers. This first type here is called structural isomers. The second type right down here shows geometric isomers. And the third type is enantiomers, or these are sometimes called optical isomers. Let's start with the structural isomers. Well, first, to be isomer, they need to have the same molecular formula. So let's ensure that that is correct. 1, 2, 3, 4 carbons, so C4. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogens, C4H10. 1, 2, 3, 4, same here, 4 carbons, and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogens. So these have the same molecular formula, they have the same atoms, same number, same ratio, but they're obviously different. What makes them different is that they have different bonding partners. These are arranged differently, they are covalently bonded to different atoms. So the difference between a pair of structural isomers is that one isomer has different bonding partners than the other. Okay, different bonding partners. Same atoms arranged differently in terms of covalent bonding. Looking at this molecule on the left, this is butane. This right here is butane. Whereas this molecule is called isobutane. And you'll notice that butane just has this linear arrangement with the carbon skeleton, 1, 2, 3, 4 in a row. Whereas looking at isobutane, there are 3 carbons, and the middle carbon is covalently bonded to the 4th carbon. And this difference in structure gives the molecules different properties. So the properties of a molecule are determined not just by the atoms that make it up, but by the arrangement of those atoms. So that is an example of a structural isomer. Now let's look at geometric isomers. I take a look at these and I see that, okay, I have 1, 2, 3, 4 carbon atoms, 3, 6, 7, 8 hydrogen. C4H8. Over here, 1, 2, 3, 4 carbon, 3 hydrogen, 6, 7, 8. Same formula. And then I look, and each carbon is bonded to a carbon, and then that carbon in turn to 3 hydrogens, and then a hydrogen down here, and then a CH3 group, and a hydrogen, and double bonded to carbon. And if I look over here, it's the same thing. Each carbon is double bonded to the other carbon, and to the CH3 group, and to a hydrogen. So they have the same bonding partners, they have the same atoms, but you can see that they're different. And what makes them different is the spatial arrangement of the atoms around a double bond. Double bonds are inflexible. The atoms can't just rotate around that bond. Now, if this were a single bond, this could actually rotate, and then the hydrogen could flip up here, the CH3 group could end up down here, and then these wouldn't actually be isomers. But because this double bond is inflexible, these are sort of held in this position. And you can see in this example, this molecule right here, these 2 CH3 groups are on the same side, whereas here the CH3 groups are on the different side. And what we call this is the cis isomer. If the 2 groups are on the same side in a geometric isomer, that's the cis isomer. The isomer where the 2 groups are on opposite sides is called the trans isomer. All right, so we have structural isomers, That was the first type of isomer, geometric. And then the third is enantiomer, or the other name for it is optical isomers. And the name optical isomer helps you remember what it is, because what it is a mirror image. Let's say that up here, the blue is hydrogen. And then maybe this brown is a carboxyl group, COOH. So in the center, I'd have a carbon bonded to a hydrogen, a carbon bonded to a carbon, in turn bonded to these. Maybe down here, I have what's called an amino group. This could be, so NH2 bonded. And then another atom, another group, say CH3. Okay, now, the purple in the other molecule is, again, CH3. This brown, COOH, and the green, NH2. And if you look at these, they're mirror images. And they are not superimposable, because this is in 3 dimensions, so maybe these two are coming out towards you. And it's similar to your left and right hands. Your left and right hands are mirror images of each other, and they're not superimposable. And it's the same idea here. And in fact, we call these left and right-handed molecules, and they're known often as L and D enantiomers. L is Latin stands for the Latin levo, which is left, and then D from the Latin dextro, or right. So optical isomers are left and right-handed forms of a molecule. And this difference is extremely important in science and in biology. For example, in pharmacology, it matters a lot sometimes which enantiomer you're working with. There are some medications where one of the enantiomers is very effective and the other is useless. Or both may have a use, but they're different. Or one may even be harmful. Certain medications are a mix of the L and D enantiomers, and one enantiomer is useful and the harmless one is in there as well, doesn't cause any problems, but doesn't help either. For Parkinson's disease, one treatment is a medication called L-DOPA. L-DOPA is the L enantiomer, and it's effective in treating Parkinson's, whereas the D form of this molecule is not. The next concept we're going to cover before we get on to some of the large molecules that you'll be working with in biology is that of functional groups. You've already seen a couple of functional groups when I just wrote out the COOH, that's a carboxyl group, NH2 is an amino group. Now we're going to go ahead and treat these formally. Groups of atoms that are especially important in determining a molecule's behavior are called functional groups. And by determining its behavior, these are parts of the molecule that take part in chemical reactions. So they determine what types of chemical reactions a molecule will participate in. And they also help to determine the shape or structure of the molecule. And there's certain ones that you'll see over and over, so you should become familiar with these and recognize them when you see them. So recall that hydrocarbons contain only carbon and hydrogen. However, in many molecules, one or more of these hydrogens is replaced by a functional group. So let's go over some of the common functional groups. The first one is called an amino group. And as its name suggests, you'll see these in amino acids. So this is NH2. And this line here indicates a bond. So this would be bonded to the carbon skeleton. So this is the functional group, and it's bonded to the carbon skeleton. Sometimes one of the carbons in the carbon skeleton is part of the functional group, and we'll see that in a second. But for now, the amino group, again, is found on amino acids, and it can actually act as a base. It can pick up a hydrogen ion and become NH3 plus in solution. Okay, so that is amino group. The second group, which I already mentioned in the last slide, is a carboxyl group. This carboxyl group is COOH, and this carbon could be part of a longer carbon skeleton. Carboxyl groups are found in organic acids, such as acetic acid. It has a carboxyl group. Acetic acid is found in vinegar. It's what makes vinegar acidic. In solution, a hydrogen ion could be lost, and then this could become COO negative, COO minus, and then a lost hydrogen. These are also found on amino acids. The next group, OH, is called a hydroxyl group. Hydroxyl groups are found on alcohols, such as ethanol. That all ending tells you it's an alcohol. And these are polar. These are polar because of the preference for the electron pair to this oxygen, the more electronegative oxygen compared to the hydrogen. So the bond between the oxygen and the hydrogen, the electron pair favors the more electronegative oxygen. Right here we have CH3 group. This is called a methyl group. CH3 or a methyl group is found on what's called methylated compounds, and it could be attached to a carbon or it could be attached to another atom actually. Here we have what's called a carbonyl group. And there's two subsets of how we name molecules containing these groups, depending on where this group is within the larger molecule. So if, let's say, we had a carbonyl group right here, embedded within the carbon skeleton, this would be called a ketone. So if the carbonyl group is in the middle within the carbon skeleton, it's called a ketone. The other possibility is that this group is on the end. So here, the end carbon contains the carbonyl group, and this is what we call an aldehyde. So again, there's two breakdowns for carbonyl groups. Ketones, the carbonyl group is within the carbon skeleton. If it's on the end, it's an aldehyde. An example here would be acetone. Aldehyde would be something like formaldehyde. Finally, we get to phosphate group. And you're going to see phosphate groups throughout biology. For example, cell membranes contain a phosphate group. This is a phosphorus bonded to 4 oxygen atoms. And one of these oxygen atoms is bonded to the carbon skeleton. You'll notice that this is a charged molecule, that 2 of these oxygens have a negative charge, and therefore it is hydrophilic. All right, now that we've talked about the basics of organic chemistry, we're going to go on and talk about the 4 major classes of organic compounds that we're going to be discussing and focusing on in biology. These 4 groups include carbohydrates, lipids, nucleic acids, and proteins. In this lecture, in addition to covering the basics of organic compounds, we're going to be going over carbohydrates and lipids. Nucleic acids and proteins are covered separately in the following lecture. Carbohydrates are macromolecules. Macromolecules, just like their name tells you, macro and molecule, are large molecules. These are a vital source of energy for cells, and they have the molecular formula C to some X, H2O times to Y. And so some multiple of H2O, various multiples of that. What this is saying is that hydrogen and oxygen are found in a 2-to-1 ratio. That's what this is actually saying. So hydrogen to oxygen, the ratio is 2-to-1. As I mentioned, carbohydrates are an important energy source. They're found in many foods, such as breads, rice, potatoes. These are all rich in carbohydrates, and you'll often hear people talk about simple carbs versus complex carbohydrates, and we're going to talk about that as well, and that has to do with the structure of the carbohydrates. Carbohydrates are sugars, and they can actually be categorized as monosaccharides, disaccharides, and polysaccharides. Starting out with just the simple situation, monosaccharides. Mono meaning one, saccharide meaning sugar. Monosaccharides have the formula CH2O N. So some multiple of CH2O, some multiple of that, where N is usually either 3, 5, or 6. If N is 3, you'll end up with C3H6O3. And if you have a 3-carbon sugar like this, it's known as a triose. 3-carbon sugar is a triose. A 5-carbon sugar is called a pentose. And a 6-carbon sugar is called a hexose sugar. So let's go ahead and look at a couple different types of monosaccharides, glucose and fructose. So glucose is the main form in which you'll find sugar stored in animals. Fructose is the type of sugar that you'll find in fruits. Glucose is produced by plants via photosynthesis, and then it can be broken down by cells to release the energy to fuel various cellular processes. Glucose and fructose have the molecular formula C6H12O6, and you should recognize this. This formula will come up, and you should recognize it as a formula for this particular type of monosaccharide, which is a hexose sugar. So let's go ahead and look at the two forms. You'll see that there's a linear form and a circular form. And the reason for this is that in aqueous solution, sugars often exist in their ring form. So this is the same sugar. These two are the same, and these two are the same, but this is just the linear form. And then in aqueous solution, it would form this ring, so you'll often see these molecules written in their ring form. And to keep track of things, we number the carbons, and we start at the top. 1, 2, 3, 4, 5, 6, and then we can refer to groups on the different carbons based on their number. 2 is here, 3, this is 4, 5, and 6. So these are 6-carbon sugars. These are hexoses. Now, when this turns into a ring, we need to keep track and see that where all these carbons go. And again, we number them 1, 2, 3, 4, 5, and 6. And we can do the same over here with fructose. So all it is that this forms a ring, and you can see the 6 sticking up right here. Same thing with fructose. Now, another thing that I mentioned before is the location of the carbonyl group, the C double bonded to O. Looking first at glucose, you'll see that this C double bond O, H, right here, this carbonyl group is on the end. And recall that when it's on the end, we call that an aldehyde. So this is actually what we call an aldehyde sugar or an aldose sugar. So glucose is an aldose sugar. Fructose, the carbonyl group is here. It's embedded within the carbon skeleton. So this is what we call a ketose or ketone sugar, so ketose sugar. And again, that's based on the location of this C double bond O. On the end in glucose, embedded within the carbon skeleton on fructose. So those were monosaccharides. Monosaccharides means that there's just one sugar. There's just one subunit. Monosaccharides can join together to form a disaccharide. Disaccharide, just like the name suggests, 2 sugar subunits. These are both known as simple sugars. If you have more than 2 subunits, it's called a polysaccharide, and those are complex carbohydrates. So simple carbs or simple carbohydrates, simple sugars, and then polysaccharides are known as complex carbohydrates. The formation of disaccharides or polysaccharides occurs when two monosaccharides join through what's called a dehydration reaction. Dehydration means you lose water, and that's exactly what happens here. So if you looked at just two glucose molecules, so this was a single glucose molecule and then a second glucose molecule, they bond together through this dehydration or condensation reaction. So before they were bonded together, what you would have had is on this molecule, a hydrogen up here, the hydroxyl group right here. And each of these corners, each of these points, that's a carbon, this is a carbon, this is a carbon, this is a carbon, this is a carbon. For simplicity, we often don't write those out. In the ring form, it's just convention that each of these is a carbon. So you had a carbon here, and it's bonded to the oxygen next to it, the next carbon, hydrogen, and this hydroxyl group. The same thing for the other glucose. So there was an adjacent glucose, free, not bonded to the next glucose. This is before the reaction. That looks like this. Then what happened is one of these glucose molecules, so this was glucose, free glucose, monosaccharide, this is glucose. One of these loses an OH. The other loses a hydrogen. So it loses HOH or H2O, in other words, water. That's why it's called a dehydration reaction. So with this gone, this OH, and this hydrogen gone, you're left with these two bonded together via this remaining oxygen. And that's what you see here. One of these has lost an OH, hydroxyl group. The other has lost a hydrogen. And that remaining oxygen then links two adjacent sugar molecules together. This bond is called a glycosidic bond. A glycosidic bond. So adjacent sugars are held together by a glycosidic bond formed through a condensation reaction. And that forms maltose, which has two subunits. They are both glucose. And this is a disaccharide. It's also known as a simple carbohydrate or simple sugar. Lactose would be another example. This is the sugar that's found in milk. And this is formed between glycosidic linkages between glucose, one molecule of glucose, and a second molecule of another sugar called galactose. Sometimes you'll hear people talk about lactose intolerance. They are not able to have dairy products like milk or cheese. And the reason is they lack an enzyme called lactase. So when we talk about enzymes, you'll see that A's ending tells you it's an enzyme. And here, ose tells me this is a sugar, so lactase is the enzyme that breaks down lactose. And if somebody is lacking this or they don't have very much of this, they're not able to break down this sugar, and that causes them to have gastrointestinal symptoms when they have dairy products. Okay, so what we started out with down here with the maltose was glucose, C6, H12O6. But we actually had two of those. Actually, let's move over here where there's a bit more room. So C6, H12O6. But we actually had two of these. And these came together to form C12, H22O11 plus one molecule of water. So 2 times 6, that gives you 12 carbons. 2 times 12 is 24, and we have 22 here in this disaccharide and the other 2 in the water molecule that was lost in the dehydration reaction. And then 2 times 6 is 12. We have 11 oxygens here within the disaccharide, and then 1 oxygen, again, lost as part of that water molecule. Now, sugars in plants and animals and organisms are usually stored in more complex forms than just monosaccharides and disaccharides, and then they're broken down as needed to provide energy. The formation of the glycosidic bond is through what's called this dehydration or condensation reaction. The opposite is hydrolysis. So hydrolysis comes from hydro, meaning water, and then lysis, meaning to break. So to break the glycosidic bond, hydrolysis reaction occurs. And what happens is it adds water. Water is added to the bond, and it releases this bond so that you then have two glucose molecules back. So maltose is formed through the dehydration reaction, forming a glycosidic bond, and then hydrolysis can add water and turn these into two separate glucose molecules. Sugars are actually stored in more complex forms, and those forms would be starch, usually in plants. And then in animals, what you'll see is glycogen. Sugar is stored in the form of glycogen. Looking at this starch molecule, you'll see that it's much more complex than the disaccharide, and it is in fact a complex carbohydrate. This is formed through glycosidic bonds that are joining more of these monosaccharide subunits and joining them in a branched form. So looking at starch, there's a couple different kinds of starch. One type is amylose. Another type, which is shown here, is amylopectin. So this is amylopectin, and you'll see that it's branched. Another thing to note is that the three-dimensional structure of starch is helical. We call starch and glycogen, we call these polymers. So when there's a chain formed from similar or identical subunits, these are known as polymers. So a bunch of subunits of glucose join together. They can be a linear form and then form a helix, that's amylose, can branch off and form amylopectin. Glycogen is even more highly branched than amylopectin. Again, it's a form of polysaccharide. It's a way of storing sugar in animals. And glycogen and starch can both be broken down through hydrolysis to release the energy as needed. Glycogen is found primarily in the liver and in muscle cells. Sugars are used for quick energy. So they're rapidly available, but they're also rapidly depleted. And that's why we have other forms of energy store, such as fats, which we'll talk about shortly, and proteins. In addition to being forms of energy, carbohydrates also have a structural function. So I was focusing here on starch and glycogen and the glucose subunits as forms of energy. And they're obviously important for that, but they also can be used, carbohydrates are also used to form structures in plants and animals. So if starch is an energy form, glycogen is an energy form, what are the structural forms? Well, plants contain cellulose. And we'll go into detail about cellulose in the plant lecture, but for now you should just know that this is a form of carbohydrate that's found in the cell walls of plants. Plants don't have skeletons, obviously, so for support and for protection, instead they have cell walls. And cellulose is a polymer made of glucose subunits that's the primary component of the cell wall of plants. It's different, though, than starch. It's not the same as starch. It's not the same as glycogen. It has a different three-dimensional structure. Cellulose actually are glucose monomers that hydrogen bond, and they form what's called microfibrils. So it's a fibrous type, more of a fiber-like or cable-like structure, a microfibril. Humans cannot digest cellulose. So what we call roughage is the part of the plant that passes through the human GI tract undigested, and that's the cellulose. So especially with raw fruits or raw vegetables, apples, figs, carrots, anything, these contain roughage. And although they're not digested, they are important for the GI tract because when we call them roughage, it's actually a pretty accurate name. It actually scrapes against the walls of the intestine, and that stimulates mucus secretions from the cells in the intestine. So even though cellulose is not digestible, it is an important part of the human diet. Okay, so structural forms of carbohydrates. One is cellulose, another is chitin. Chitin is found in the cell walls of fungi such as mushrooms, and it also makes up the exoskeleton of arthropods such as crustaceans, also insects. So this is found in exoskeletons, and it's also found in the cell wall of fungi. Again, we'll revisit this all in detail in later lectures, but just giving you some examples now to make this more concrete. So two functions of carbohydrates. One is as an energy store in forms such as glucose, maltose, lactose, starches, glycogen. The second, structural functions. And this could be cellulose, found in the cell walls of plants, chitin, which is found in the cell walls of fungi, as well as the exoskeletons. All right, carbohydrates were the first class of large biological molecule. The second group of large biological molecules that we'll be discussing is lipids. And these are actually a diverse group of molecules, but they're classed together because they're all nonpolar or hydrophobic. Examples of lipids are fats, oils, phospholipids, and steroids, as well as waxes. We're going to be focusing on fats, phospholipids, and steroids because these are the most important in terms of biology. If you glance down here at a typical fat, this is a triglyceride or fat, you'll see that it has these large regions of hydrocarbons. So these long hydrocarbon chains are regions that are nonpolar. So I mentioned that earlier in this lecture that hydrocarbons are nonpolar and nonpolar molecules contain regions of hydrocarbons. So let's go ahead and see how a triglyceride molecule is built. It's built from one glycerol and 3 fatty acid molecules. So one glycerol plus 3 fatty acid molecules equals 1 triglyceride. Glycerol is an alcohol, and each glycerol then bonds with the 3 fatty acids. And you can see how these, where these are linked. If you look first at the glycerol molecule and then this OH group, and then you'll see CH2O, and then it's bonded to the first fatty acid, the second fatty acid, and the third fatty acid. And this is, again, a dehydration reaction. So if you look here, you'll see OH, and then C, double bond O, OH. I look over here, there's 2 oxygens, and this carbon, and a carbon here. What's happened is, through the dehydration reaction, 2 hydrogens and 1 oxygen have been lost. So the dehydration reaction, loss of H2O, and then the glycerol molecule ends up bonded to the fatty acid via this oxygen. This type of linkage is called an ester linkage. An ester linkage is a bond between a hydroxyl group and a carboxyl group. So a bond between these 2 types of functional groups that's formed is called an ester linkage. So here we have 3 ester linkages between 1 glycerol and 3 fatty acid molecules. And so you can see the formation of that between these 2 molecules to here. Adding 2 more groups gives you the triglyceride, and the tri meaning 3. So 3 fatty acid chains on the 1 glycerol molecule. Fats can be divided up into 2 different categories, and you've probably heard these categories or read them on nutrition labels: saturated and unsaturated. When we say that a fat is saturated, we mean that the carbons are saturated with hydrogens. Think of it that way. Another way of looking at it is there are no double bonds between the carbons. So this is a saturated fat shown here because all the carbons are held together by single bonds, and each carbon is saturated with hydrogens. It's holding as many hydrogens as it can. It's bonded to as many hydrogens as it can be. Unsaturated, there's one or more double bonds between the carbons. For example, if I inserted a double bond here, I would have to then get rid of this hydrogen. This carbon is no longer fully saturated with hydrogen. It has a double bond to the adjacent carbon. You'll often hear people say, oh, saturated fats are the bad ones and unsaturated are the good ones. So let's talk about some differences between these two types of fats. Saturated fats have no double bonds, and they often come from animals. They're animal fats. They are solid at room temperature, and the molecules that comprise them pack together tightly. And that has to do with the fact that there's just single bonds. An example would be butter. Unsaturated fats usually come from plants. They are liquid at room temperature, and they don't pack together as tightly. The reason for that is this double bond creates a kink. You'll often see the fatty acid tails. So if this is the glycerol, you'll sometimes see these fatty acid tails shown as zigzags, like this. That's their structure. And so on. Now, when there's a double bond, it introduces an even larger kink. So it would go out to the side like that. For example, this might be a saturated fat, and then if there was a double bond, it actually kinks the molecule so that it's not just a straight tail like this. And you can see that if this is kinked, these could not pack together as tightly. They couldn't pack together with adjacent fat molecules. So this double bond causes a kink in the fatty acid tails that doesn't allow them to pack together as tightly, and that's also why they stay liquid at room temperature. They don't solidify because they're not packed together as well. A diet that's high in saturated fat increases a person's risk of atherosclerosis. And atherosclerosis is the formation of plaques in the blood vessels, and these plaques decrease the flow of blood through the vessels, and they can result in heart disease. So often unsaturated fats are thought of as the healthier fats. An example would be canola oil. Canola oil is mostly unsaturated fat, whereas butter is mostly saturated fat. Now, fats are a way for organisms to store energy. I mentioned that animals can store glucose as glycogen, and then they can break it down. But that gets rapidly depleted. For longer-term storage, a very efficient way to store energy is as fat. So adipose tissue serves as a way to store energy, and it also serves as insulation and protection. So those are some functions of lipids. Another function of lipids, in addition to fats, would be to form phospholipids. So we talked about one type of lipid, which is fat. The second type we're going to talk about is phospholipids. Cell membranes are made up of phospholipids. And let's go ahead and look at the structure. Here, we have the glycerol molecule again. And we have fatty acid tails, but this time we only have two. Instead, this third carbon, instead of being bonded to a third fatty acid, it's bonded to a phosphate group. This phosphate can in turn be bonded to another functional group, and that'll determine which type of phospholipid that you have. But the basics for phospholipids are glycerol, two fatty acid tails, and a phosphate group. The phosphate plus the glycerol, this region is hydrophilic. So we say that phospholipids have a hydrophilic head, and then we look at the fatty acid tails, and we say that they have hydrophobic tails. Because there's one section that's hydrophilic and another that's hydrophobic, we say that this molecule is amphipathic. An amphipathic molecule has a hydrophobic region and a hydrophilic region. Because of this amphipathic nature, phospholipids self-aggregate into bilayers. And they do this in order to shield these hydrophobic portions from water. So what you end up with is phospholipid bilayers where the hydrophilic heads face outward and the hydrophobic tails are sequestered inside. So this is the hydrophobic portion and it's sequestered inside from the water. The interesting thing about this as well, the structure, is that it can serve as a barrier, and it does serve as a barrier, so that the cell membrane only allows certain substances to pass through. And in particular, hydrophobic substances will pass much more easily through the cell membrane because of this phospholipid bilayer's nonpolar central region. So hydrophobic molecules tend to pass through the cell membrane more easily than hydrophilic molecules. This is an introduction of phospholipids, and we're going to do a lecture later on in the course on cell membranes and talk more about this structure as well as about substances that pass through the cell membrane easily and those that do not. All right, so far we've discussed fats and phospholipids, which are both types of lipids. The third type of lipid we're going to discuss is steroids. Steroids are lipids that contain a carbon skeleton of 4 fused rings. This ring structure, this basic ring structure, is called cholesterol. And it's the building block for other steroids. Hormones are substances that allow cells to communicate with and affect cells a distance away. For example, a hormone would be estrogen or testosterone. These are both hormones. Many hormones are steroids. These two are steroids. So estrogen and testosterone are examples of steroid hormones. That means that they have this basic 4 fused ring structure, but there are different functional groups attached. The bonding's a little bit different. So alterations are made to these that give each one a unique structure and very different function and properties. And if you think about it, you can see that this is going to be pretty nonpolar. It's going to be hydrophobic, and that's actually very helpful for a hormone because it allows them to pass into the cell. Again, we're going to revisit this topic later on when we talk about cell membranes and cell communication, but for right now, you just need to know that hormones are often steroids, and that steroids are based on a cholesterol ring, and they are nonpolar. All right, today we've discussed 2 of the 4 classes of large biomolecules. We've discussed carbohydrates and lipids, and we're going to continue on in the next lecture to talk about nucleic acids and proteins. But before we do, let's do a few examples to reinforce your knowledge. Example 1. What type of isomer is each of the pairs of molecules shown below? Remember that isomers have the same molecular formula. So let's just double check and see that that's true. I have 2 carbons here, 2 bromines, and 2 hydrogens. On the other molecule, I also have 2 carbons, 2 bromines, 2 hydrogens. Same molecular formula. The first type of isomer is a structural isomer. So let's see if they have the same bonding partners. Each carbon is double bonded to another carbon on both of these, and then each carbon is bonded to bromine and hydrogen. Bromine, hydrogen. Same here. But they're not the same geometrically. They differ in their spatial arrangement. So these are not structural isomers since they have the same bonding partners. In fact, they are geometric isomers. And one clue to this is the double bond. This inflexible double bond holds these molecules in a certain conformation where the 2 bromine molecules are on one side in this isomer, so this would be the cis isomer, and they're on the opposite sides in the other isomer, and this is known as the trans isomer. So that's my first set of molecules. The second set, I'm going to double check and make sure the molecular formulas are the same. 1, 2, 3 carbons, 1, 2, 3, 4, 5, 6, 7, 8 hydrogens. 3 carbons, 8 hydrogens, 1 oxygen. Over here, 1, 2, 3 carbons. 1, 2, 3, 4, 5, 6, 7, 8 hydrogens, 1 oxygen. So these have the same molecular formula. Next thing to check, are the bonding partners the same? Here I have a linear arrangement of carbon, 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 and then oxygen. Here I have the 3 carbons, but instead of the oxygen being bonded to a carbon on the end, the oxygen is bonded to a middle carbon. Therefore, the covalent bonding is different, and these are in fact structural isomers. These are both forms of propanolol. This is actually propyl alcohol, while this one is isopropyl alcohol. And again, these different isomers are going to have different properties. So the first example was geometric isomer. The second is an example of structural isomers. Second example. Identify the functional groups on the molecules below. So let's go ahead and just look at the linear. This shows glucose in both its forms. We can look at it in either, but I'm going to look at the linear. And this is actually an amino acid. So let's look at all 3 of these and just see what we can identify. One thing we can identify here is a hydroxyl group. Here on the end, it has a carbonyl group. And remember that when it's on the end like this, we often call it an aldehyde. If it was in the middle, we would call it a ketone group. Over here, I have COOH right here. So this group is a carboxyl. The carbonyl is C double bond OH. So I've identified hydroxyl, carbonyl, carboxyl, and then I have an NH2 here. NH2 is called an amino group. What do I have here? CH3, I have 2 of these, and these are methyl groups. Okay. So here, some of the various functional groups that I've identified: carbonyl, particularly an aldehyde, hydroxyl, aminyl, carboxyl, and methyl. Example 3. The structural formula for galactose, a sugar, is shown below. Based on the number of carbons, what type of sugar is galactose? Is it a ketose sugar or an aldehyde sugar? Okay, so this is galactose. 1, 2, 3, 4, 5, 6 carbons. Therefore, this is a hexose sugar. To determine if it's a ketose or aldehyde sugar, I need to look for the carbonyl group, and that's right here. It's on the end carbon. Therefore, this is an aldehyde sugar. So galactose is a hexose sugar, and it's an aldehyde sugar. Example 4. What class of molecules does this molecule belong to? Why? Well, you probably immediately notice this characteristic structure of 4 fused rings, and this molecule, therefore, is based on cholesterol. Therefore, it is a steroid. Notice, though, that the bonding, the functional groups are a bit different than on cholesterol. And in fact, this happens to be estradiol, which is a form of estrogen. So you can see that changing this structure just a bit gives it very different function and unique properties. That concludes this lecture on organic compounds at educator.com, and I'll see you again next time.